So let's talk about proteins for a minute. Proteins. When you think about proteins, they're pretty much the workhorses of the cell. They do everything for us, and not even the cell, for just organisms in general. They do everything for us. And usually when I think of proteins, I think of two big things. I think of structure and enzymatic reactions. And those are the two biggest ones, but they're not the only ones by any stretch of the imagination. So if you look, you got several right on the top of your handout there. So if you look, enzymatic reactions, all enzymes are proteins. Not all proteins are enzymes. There's other types, but all enzymes are proteins. They catalyze reactions, really super important. So past that, they provide structure. So they provide structure. For those of you that still have hair, the structure of your hair, so it comes from a protein. So movement, actin and myosin. So actin and myosin, one, in your actual muscles, but two, inside the cell, they play a role for movement and transport and things of this sort inside the cell itself. So for defense, your antibodies are proteins. Regulation. So on the surface of cells, receptors and things that bind oftentimes receptors, and the signaling proteins as well, all convey signals from outside the cell into the cell. So regulation as well. And then transport. So transport, notice you got a cell membrane on most cells. So if something wants to get in, there's a few things that actually can diffuse through the cell membrane, but most things don't. And they need some sort of channel protein that gives them safe passage through into or out of the cell. So proteins do a whole lot of stuff. So just summarize kind of there. You might just want to kind of make that list and memorize it real quick because that makes a great multiple choice question as well. Cool. So we'll talk about three classes of proteins here. So we're going to talk about three types, and one of them we already hinted out. So that's your globular proteins, proteins. These are typically the proteins that are in the cytosol, in the cytoplasm. They're just dissolved in the cytosol, doing their job. Because they're in the cytoplasm, which is what medium again? What's the solvent? Water. So that's why they have the structure we talked about earlier. So they usually form some sort of crazy compact structure, and there's lots of variability in that. But the key is they put all the non-polar amino acids, for the most part, on the inside, and then all the polar ones face out. So all the polar ones are chilling with water, and all the non-polar ones are just chilling with each other, but specifically not with water. Cool. Your next class, your membrane proteins. So I'm assuming you guys have had a biology class. You usually kind of have to before biochemistry. So cell membrane, what is it typically made of? Lipid bilayer, phospholipid bilayer. So you usually get a polar head group with two tails, right? And these things just line up next to each other and form a big, huge bilayer. There's half the bilayer. There's the other half. Cool, so the head groups are polar, but the interior of the bilayer itself, these are big, long carbon chains, so fatty acids, if you will. So, and being big, long, just plain old hydrocarbon chains, they're nonpolar. So the interior is nonpolar, but the outside barrier itself is polar. And so if you want something to get transported across, like let's say I want to transport, I don't know, calcium ions or something like that across this membrane. Calcium says, hey, how's it going? You phosphate groups, you look pretty cool. And then it tries to go in and says, oh my gosh, you guys are terrible. You guys hate me and I hate you. So calcium ions, or any ions in general, don't like the interior. And most ions don't just diffuse across a membrane. They got to have some sort of channel protein or some sort of process to either facilitate it or actively transport it in and out of the cell. And so in this case, if I wanted to put something in here, so what I would do is get a protein, and this protein would actually embed itself into the membrane itself and often form a hollow channel. A hollow channel. Now, in this case, all the proteins that face out here, what do they see? They see the lipid part of the lipid bilayer. Guess what kind of amino acids those should be? Nonpolar, nonpolar. Sweet, but then all the ones that kind of face the open channel here where calcium or something like that might be going, Those are going to be your polar ones. Sweet. And that's how a membrane protein's fairly different in construction than your globulars. So your last class of protein that we're going to talk about anyways is your fibrous proteins. So fibrous proteins are known for forming big, long fibers. So what was the big example you guys learned about in class? Collagen? Collagen, maybe? So keratin might be another example, but collagen. Collagen forms a triple helix. It's like making a rope out of 3 separate strands and just twisting them around each other. It forms a triple helix. So typically nonpolar amino acids in a very repetitive way for your fibrous proteins. So hydrophobic or nonpolar residues and very repetitive in its sequence. That's what you should know for that. Cool. Those are your 3 classes. So, and the way they're kind of designed, especially for the globular membrane, the way they're designed really makes sense for what they're supposed to do. So protein denaturation. Protein denaturation. Just a quick vocab word, couple things about it, and we'll move on. But denaturation is when a folded protein unfolds. So in this denaturation process, one, it can be affected by changes in pH. If your pH changes from like 7.4 physiological pH, even if it doesn't even change by a significant amount, even if it changes a little, you might have proteins that misfold or at least unfold a little bit in some cases. And if they misfold or unfold, the structure of a protein totally gives you its function. And so if a certain protein is an enzyme responsible for a chemical reaction, if it unfolds a little bit, it was the way it was folded that often gives it its ability to function as it is. And you unfold it, you might just have a worthless protein at that point, a worthless enzyme. And so this denaturation process is a really big issue. So things like pH, temperature, salt concentrations, detergents, organic solvents, all these kind of things. I think I'll probably put a couple others on your sheet, huh? Reducing agents. Why would a reducing agent cause this? Breaks the disulfide bridges. So, or even mechanical stress. We actually used to have some fun in the lab. And we would tether one end of a protein to a mica surface. And we put the other end on what's called a cantilever. And we'd stretch the protein out and play with it a little bit. And we could actually do one at a time, one protein at a time. It was pretty cool stuff at the time. And so mechanical stress can also lead to this. And we were actually studying protein folding a little bit and protein aggregation and amyloid stuff and things of that sort. But if proteins do this, they often lose their function and they are garbage. They either got to be refolded or just degraded back down so that we can reuse the amino acids in the body and stuff. So if this gets to be too big of a problem, so notice if all of a sudden you have all these proteins that have exposed nonpolar regions. So and if I see Keon over there and he's misfolded, he's denatured, and he's got exposed nonpolar regions, and I've got exposed nonpolar regions, I'm like, dude, we should hang out because this water thing sucks. And we'll get some hydrophobic interactions, and so you start forming these protein plaques, which is a really big problem, like in Alzheimer's or Parkinson's.